In this video, we'll be looking at epithelial tissue. Now, epithelial tissue is one of the four main tissue types. Now, like all the four main tissue types, they are subdivided into various types. So not surprisingly, with epithelial tissue, we're going to see that we're going to have different names and different looks to it depending on where it's located and what its function is. And remember that concept of form and function go hand to hand. Now, where do we find epithelial tissue? Now, epithelial tissue we really find on internal and external body surfaces. And really, what's its job? It acts as a barrier between the inside of you and the outside of you, basically between the body and the external environment. So it lines organs. You find it in fluid-filled cavities. Your skin are examples. The lining of your oral cavity, your nasal cavity, your GI tract. And if we start to look at the functions, we can sort of break it down into some major categories. Protection. So it helps protect the underlying tissue from injury, and that injury could be mechanical, a stress placed upon it, a force, or thermal. The other thing too is it really is a barrier, and although it's not the same part of the immune system that T cells and B cells and all those other white blood cells that we're talking about, we'll talk about in the immune chapter, this is the first line of defense for your body against microorganisms and viruses and fungi and all those other potentially infectious agents. And so it does act as a first line of defense. It's a physical barrier that's part of the immune defenses. As I said, it's a physical barrier and it prevents the invasion of microorganisms. Now, the other interesting thing is often when we see epithelial tissue, below epithelial tissue will be tissue which will have cells that are part of the immune system. Also secretion. So epithelial tissues are a very important component of what we call glands. So hormones, oils, etc. Really, we often see epithelial tissue as a main component of that. Transport to other tissues in that selectively permeable membranes. So substances are able to cross these barriers either passively or actively and enter other tissues. So it could act as a barrier, but not a complete barrier, that some things may have the ability to cross it. So it helps to control potentially or regulate what can move from the outside world to inside you. And let's not forget sensation. That with many epithelial tissues, we have different receptors. And of course, it's really a great benefit to have that concept of sensation. So we often see rich nerve supply, and that helps us to detect changes in the internal-external environment. One example, our taste buds are a very specialized sensory epithelial cell. Now, you could spend some time, read the book, look at this, but one of the big problems people first face when they're dealing with histology is, what the heck am I looking at? It all kind of looks like this bluey, purpley, pinky mess. When you're first facing histology, you really have to enter it knowing what should a given tissue look like. It's almost like you should have a checklist in your mind that what would epithelial cells look like, or epithelial tissue. Tightly packed cells, very little matrix. So you want to keep a running list. Now, often when you're looking at histological sections, and you experience this when you're looking at cells in the previous cell lab, is that the sections will often show more than just one tissue type, because we actually have two different tissue types here. And you can see that this area looks quite a bit different than this area. And with a little bit of expertise, you're going to be able to start to figure out what type of tissue am I looking at based on the criteria. Up here, by the way, those are darkly stained nuclei. You can see these cells are very tightly packed together up here. Down here, the purple nuclei are basically where the cells are. And then we have a lot of wavy fibers. So these are two distinct tissues. By the way, maybe you can even recognize these. These are little red discs, anucleated. Yeah, they're red blood cells. And actually, this is a type of epithelial tissue that's lining the capillary or the blood vessel. So At first, as I said, it's really daunting because you're looking at this sort of mess, what looks like a mess. But after a while, if you know the key features, and that's the big thing, key identifying features, that will help you start to understand and to be able to recognize what you're looking at. Here they're using an example of the esophagus, and I'm not going to walk through it in great detail, but you can actually see these are huge bundles of collagen in there. And later on, when we get to connective tissue, we'll talk about exactly what type of tissue that is. You see the collagen fibers are sort of, they're not organized in a perfect way. That they seem to be going off in slightly different directions. And that can be a key clue as to what type of tissue you're looking at. Okay, so let's get into looking at epithelia itself. And as I said, epithelia is all about tightly packed cells. And so that is the hallmark of epithelial. A lot of cells, very, very little visible matrix. And we have tight junctions and desmosomes that hold it together. So we can see here's a very classic looking epithelium. You see how tightly packed the cells are. There's no visible matrix in between there. And really that is one of the biggest clues. Now, just a little side note is that whenever we start to see epithelium, we'll learn this, is that we usually start to run into tissues that are right below it. Because remember, epithelium faces the outside world or cavities, or it faces inside cavities within you. And we'll see that it's quite common that epithelia will have a basement membrane below it. And we'll come back and talk about that. So, as I said, tightly packed cells, also impermeable and resistant to physical stresses and mechanical injury. There's a lot of tight junctions which help to hold it together. Also desmosomes. Remember, desmosomes give you a lot of strength to hold cells together. Now, the other thing about, let's go back here for a second. Look how tightly packed it is. There's really no room for blood vessels to move through there. So not surprisingly, epithelial tissue is avascular. Just a reminder, the letter A in front of a word usually tells you no or lack of. So epithelial tissues are avascular, lack blood vessels. Where are you going? Well, they're living tissues. Where do they get their oxygen from? And they get their oxygen from, not surprisingly, is typically when you see an avascular tissue like epithelium. We're going to have tissue below that will be vascularized. So we're looking at a process of diffusion, which means that epithelial tissue can't be too thick because you can't be too far away from your blood supply and your nutrient supply. So as I said, it's avascular, but it has to get its nutrients and oxygen from deeper tissues. So that really limits the thickness of it. Now, usually when we're looking at epithelial tissue, is we have a thin basement membrane. And the basement membrane has two parts to it. Okay, the basal lamina, and so the extracellular matrix synthesized by the epithelial cells, and this is collagen fibers and ground substance. And then we have something called the reticular lamina, which is made by the underlying connective tissue, which has reticular fibers and ground substance. And these two layers, what do? They help to glue or attach the epithelial tissue to the underlying connective tissue. And that's something we see quite often is we have epithelial tissue, then a basement membrane, and below the basement membrane is typically some type of connective tissue. All right, and let's just hop back here. By the way, let's just finish this off actually. So we have the two layers, the basal lamina and the reticular lamina. And as I said, they glue themselves together to help to kind of glue the epithelial to the connective tissue. And of course, there's underlying blood vessels below that, and it's a barrier between the epithelial and underlying tissues. So let's go back to the diagram. We can see it here. So here's our avascular epithelium, or epithelial tissue. We have a basement membrane, and the basement membrane is made of the basal lamina and the reticular lamina. And the basal lamina is really extracellular matrix that was produced by the above epithelium. And the reticular lamina is contributed to by the underlying connective tissue. Now, when we're looking at epithelia, we should start to think about how are we going to classify it and how are we going to give it names. Well, there's a couple of variables we can look at, and one is how many cell layers are there? And let's start with that. That in some cases, the cell layers are we only have one cell layer thick. In other epithelia, we actually have multiple cell layers. You can see all the individual cell layers. Okay? So if it's only one cell layer thick, I'm going to call it simple epithelia. If it's more than one, I'm going to call it stratified. And we're going to see this name show up in any description of epithelia. It's either going to be a simple or it's going to be a stratified. The next thing that we're going to see is what's the second name that we're going to give it, because epithelia usually has two names, like a first name and a last name almost, and it's based on the shape. Okay, so a squamous is a flattened type cell. A cuboidal cell looks like a cube, and a columnar, not surprisingly, looks like a column or columnar. So we can also include these in their names. And once again, when you look at the names, part of the name is going to come from the number of layers of cells. Part of the name is going to come from the shape of the cells. Now, we're going to ease out there because I'm going to come back and start talking about the different types of epithelium in more detail. So we'll pick that up in the next video.